This meeting is being recorded.
Yes.
Now start.
Shall I read it?
Yeah.
Pardon?
Yes, yes, read out.
Oh, okay. It says, a young, what's the story? Rosalind Franklin, 1920 to 1958. A young Rosalind Franklin wearing a short bib and a collared sleeved shirt peers down into a microscope.
Yeah, yeah. Which is
The
shown in
race
your
is to
figure, no?
discover, pardon?
It is shown in your figure.
Yes.
Yeah.
The race is to discover the structure of DNA consumed scientists in the 1950s, but it was work of one of women, Rosalind Franklin, that proved instrumental in uncovering the double helix.
Yeah. It is the structure of the DNA, which is a double helix strand. Okay.
Yes, so he was
The
like
structure
she
of
was
the
looking for
DNA.
that.
Yeah, yeah. This DNA structure is discovered by her, right?
Okay,
And in the microscope,
right.
she saw that.
Okay,
Yes. Next.
so Franklin held a PhD in physical chemistry from Cambridge University and worked on X-ray cytography.
Yeah.
Cytography. She successfully photographed the structure of DNA on a machine she refined after 100 hours of X-ray exposure. Her colleague Maurice Wilkins gave the photograph to James Watson and Francis Crick without Franklin's permission. When Watson saw picture, he said, "My jaw fell open and my pulse began to race."
Yeah, yeah. Understood? That one,
Yes,
yeah. That PhD she has done in the physical chemistry from that university, Cambridge. And the
yeah.
structure of the DNA photographs is shared by her colleague. And those who know about
Maurice
that, they just
Wilkins.
surprised to see
Yeah,
that
so like
picture.
she gave the
Because
photograph
without her
without her
permission,
permission.
yeah, yeah, yeah.
Yeah.
Now,
Watson
next.
and Watson and Crick used Franklin work to publish a revolutionary 1953 article in the
Yeah,
journal Nature.
ultimately,
He said something, miss, I didn't understand.
yeah, yeah.
Okay, so and ultimately winning them the Nobel Prize, an honor they shared with Wilkins. Unfortunately, Franklin passed away at the age of 37 without receiving recognition for her contribution to science.
Okay. Franklin passed
Why
away
does
before that, no?
yes, so like she couldn't even find it out
Yeah.
that like she got a Nobel Prize.
Yes, yes, yes. But she discovered that. So that's the history
Yeah.
about this DNA strand and the discovery of the DNA structure, right?
Yes.
Then next question is,
Right.
why does this matter? Studying
Studying
cells.
cells. Oh yeah.
Yeah. Okay, read out.
Okay, so studying cells help us understand how organism, including human function. After all, our body is made up of trillions of cells. By learning about cells, we came to understand how we can protect cells to prevent infection and other harmful effects, observe cells to diagnose disease, treat cells to heal illness, stop harming cells through our choices and action. Sound familiar? Cell biology studies affect a wide range of disease and other basic cellular questions, with new tools leading the way. Cells can turn into various other types of cells. Scientists believe that they can be useful for treating and understanding disease.
Now, in this, that the how the study of cell is important and how the body is made up of. Body is made up of the trillion of cells. Means much large number of cells are there.
Sorry, Miss, I didn't quite get it.
Very large number of cells are there in the body.
Yes, it's like
It's
in trillion
yeah,
cells.
it's a trillions of cells. So what's the function of about these cells? They protect us from the infections and the harmful effects. And with the help of the cells, we can diagnose many diseases. Like many diseases are there in which the platelets counts are low, or the eosinophil cells are higher, which is just 0 to 4% in our blood cells, right? So with the help of these type of cells, we can diagnose many diseases and we should treat it. If there is a low platelets, then we can give the diet to the patient to raise the platelet count, right?
Right. So like it kind
So
of helps us to like understand the disease. So
yeah,
that's
and
why
then
doctor can
to
do a bit
prevent
more.
it and to heal it also, to heal that illness, we should know about these cells, right?
Right.
And stop
Right.
harming the cells through our choices and actions. Like if someone doesn't know about her platelet count, but she is taking other diet which also lowers down the platelets, indirectly she destroy their own cells, no? Right?
Yes.
So when she get know about this that she has a low count, then she stop eating those things which are harmful for her, and start eating those things which is helpful to increase the platelets. So it will also help in this, like in the treatment, to avoid it and also
Okay.
to increase it.
Okay. Yeah.
Understood? Now next is curricular concepts.
It's
It's
something
about
that miss
the
we don't do
student.
in school. It's like in every
Uh-huh.
knowledge organizer.
Yeah. Now next page number 7.
It is it is sick. I mean it shows mine and six. It's it's something I can't pronounce most clearly. I'm terrible at pronouncing things.
Yeah. On the 7 page number, we have to revise the cycle. Below
Um
that, biophysicist, what they
yeah,
do,
wait.
right?
What no. It's what page are you on, miss?
Page number 7,
Okay, yes.
biophysicist, you have seen.
Yes, miss, I can see, but I just can't pronounce it clearly because I'm not right. Can't pronounce it really
It's a
nicely.
biophysicist.
Yeah.
These are
Shall
the
I read it
persons.
or you?
Okay, I
Yes.
will make up. What they do means what's the function of the or what are the purpose of the biophysicist, right? It is shown in this figure. She sees in the microscope. They help or they study the living organism by collecting, analyzing, and interpreting the data in regards to the cells and the organisms. Means biophysicist is a person who study about the living organisms by collecting, analyzing, and interpreting the data with regards to the cells or the organism. And second, analyzing the movements and signals between the cells. Because biophysicists help in the study of the cells. And
Okay.
some biophysicists work in the mathematical models also, developing the imaging equipments, developing the biomaterials. Means biophysicists is not having only one action or one purpose or only one function. They can also do work in these three categories: mathematical models, developing the biomaterials, developing the imaging equipment. They can play an important role in these fields also. So now understood biophysicists. Do you understand?
Miss, I didn't understand at the last.
Developing the biomaterial, like
Yeah.
material which is
So
used.
miss,
Yeah,
I've got a question.
yeah. Yes, speak out the
Pardon?
question.
What's the number third means? Like developing biomaterial? It's like is there a different material or something?
Material is just like we have whenever in the chemistry lab we are preparing some material or chemicals, right? And in this it is
Right.
the biomaterial. Bio means living. Living means living material. How we can make it? Like you are growing a plant.
What
It's a
biomedical?
living, na? You are growing a
Yeah.
plant.
Miss, I didn't quite understood you. Can you say it again, please?
Plants are living or not?
Pardon?
Plants.
Plants? Yeah.
Yeah, it's living or not?
It's
It's living.
I still understand what you're saying. Like,
I'm
I know
saying,
plant.
I am saying, plants are living or not? It's a question for you.
Oh, okay.
Yeah.
Okay.
So tell me, plants are they living?
Plants are living, miss.
Yes. So if we have grown a plant in our garden, then it will
So,
give us the plant after
yeah.
a few months. It will give us a plant, no? Then fruit also, right?
Yes.
So these are the
Right.
biomaterials which we study. In the same way, because cells are present everywhere, no? Plants
Yeah.
also having the cells, but meristematic tissues, lateral meristems. There are many parts. So everything is
Yes.
made up of cells. If we grow a plant, it also contains cells. So biophysicists says that
Yeah.
it develops the biomaterial, developing the biomaterial by growing the fungi or the plant. It also grows these cells, right? That person can grow any type of biomaterial for the study of organisms or the cells. Understood?
Yes, miss. Okay. Yeah.
That is the developing means for the study, for the purpose of the study, like fungi, if someone wants to study, then he will do it, no?
Yeah.
Grabbing the bread piece and take it away for the 12 hours. Then after seeing it, we can find the fungi on it, right? So in this way, they develop the biomaterials for the observations. Now, next is the what is the function of a microscope? The major function.
Microscope.
With the help of the microscope, we can see a very minute things, minute cells, minute organisms also, right? Even their bonds, even their structures, internal structures are also seen in the microscope. We can
Yeah.
say all the living organisms are made up of cells, and these cells are the building blocks of life. To observe the cells and the details, you will need to use a microscope. The microscopes magnify the object that you are looking at. Like if you want to see a piece of the onion, have you seen that in the microscope? Have you done
Miss?
peel? That is the inner peel. On the onion, there is a one thinner layer, no? Transparent layer is
Yes,
there.
miss. We have done the
It is,
experiment. It's
yeah,
like,
yeah. You have
yeah.
seen that boxes like
Yes,
structure
miss. Like, it's
options,
like a
right?
sort of like bricks, stuff like that.
Yes. Then what is that? You have seen it in the microscope, not naked eye. Like if you are see the onion in the kitchen, you can't see those cells, but you can see in the microscope. Means microscope gives us the magnifying objects. The detailed one, the bigger one, it is shown in your microscope with the help of the lens, right? And the plant cells, they can be seen using the light microscope. These two kind of microscope, simple and compound, right? And the
Right.
different types are there: light microscope, electron microscope, and too many. But after that, also many types of microscope are there. But there is, if there is two lens, then it is a compound. And there is one lens, then it is a simple microscope. So plant cells are seen in the light microscope. right? And here is also given the example of
Right.
the same microscope slide of the stained onion peels. And here's the typical method to do that, and you have also done that, and easy to understand for you also. All the living
Yes,
things,
Monsieur, we have
all
done the
living,
experiment of like skin
yeah,
of onion.
yeah. All the living things are made up of cells. These are the building blocks to observe the cell in detail. Okay. Then next line, you should be able to describe how to prepare a microscope slide of a stained onion cell. Here is the typical method. First, cut open an onion. Second, use the forceps to peel a thin layer of skin from the inside. Third, lay the layer of the epidermis on a microscope slide. Fourth,
Yeah.
add a drop of the iodine solution to the layer. Fifth, carefully place a cover slip over the layer. These bubbles should not be there in the cover slip, no? So that's why we have
Yeah.
said carefully place the cover slip. No bubble should be inside that.
Okay.
Iodine solution stains starch in the cell blue black, making the cell features easier to see. Here we have seen that bricks like the layers of that bricks. It means they are the blue black in color due to presence of the starch in it. That's why we have put the iodine solution. Otherwise, there is no demarcation in the cells when we see it. Understood? That black layer is due to the presence of
Yeah.
iodine solution and due to the presence of the starch. Because iodine, when react with the starch, it will give the blue black color. Understood?
Yes, miss. Understood.
Now, next is magnification. To work out the total magnification of the specimen, you would need to find the ID, lens magnification, and then the objective lens that you have selected. Here is an example of how to set out calculations. Eyepiece lens magnification is 10, objective lens is 20, the total magnification is 20, 200 magnification. Understood now? Is there any doubt?
No, no, not yet, miss. I am understanding it.
Okay. Next page number 11. Animals and plants are the same in
Yeah.
front of you. The major difference, major difference. In the picture, you have seen the two major differences. Tell me one by one.
What did you say, miss, at the last?
Difference between animals and plants and which is more people.
Yeah.
Tell me.
Pardon?
Major difference which is shown in the figure. Tell me between animals.
I should tell you what, miss.
I'm telling you how the question. Difference between plant cell and animal cell.
Oh, so
Shown
miss,
in your
it's like,
figure which is in front of you.
okay, so like, miss, it got one of them got like cell wall and one of them don't.
Second one, I can't
And
understand.
pardon, miss?
Second, first is cell wall, second is
Second is you could say, miss, both don't have
I don't understand.
chloroplast.
Don't have
Chloroplast, miss. Like animal cell got
chloroplast. Okay.
chloroplast.
Yeah.
Yeah.
Next, I'm saying in the figure what you have
And nucleus. No, nucleus got both of them got nucleus.
yeah.
And
Vacuole is
it's
there.
got vacuole.
Actually, yeah, big vacuole is present in the plant cell due to which the nucleus is also goes towards the periphery. But not in the
Yeah.
animal cell. In the animal cell, nucleus is always placed in the center.
Okay.
And the cell wall is the major difference. These two are just shown in the figure from very far away. We can determine it. No? Now,
Miss, I've got a question.
yeah,
Like, why only one of them got like cell wall, not another?
because animal cells, like in our human beings, we should need a cell wall or not. Because we are not attached with the environment directly. No? Our skin is present, right? it also has the 7 layers,
Right.
and in that the cells are even protected. But in the plants, they are very close to the nature and in place in the environment to face the many situations, winters, summers, rainy season,
Yeah.
many
Yeah.
type of situations they face. So they need the most protection as compared to us.
Okay.
So that's why animal cells don't have, and also there is no presence of cellulose in us. Cellulose is present in the plants by which that cell wall is made.
Okay. Yes.
In us, it is starch. There is no cellulose, it is starch in animal cells. Okay?
Okay.
Yeah. Now, next is animal cells and plant cells both contain, both having the cell membrane, cytoplasm
Yeah.
also, nucleus also, and the
Yes.
mitochondria also. Means the similarities between these two are these organelles which is present. And the plant
Yes.
cells
And like
contain cell wall, vacuole, chloroplast. It is a major
and
difference.
chloroplast.
Yeah, these
Yes.
are the major differences between these two cells. And the animal cells
Okay.
usually have an irregular shape because there is no cell wall, no fixed shape is there. But the plant cells, they have a fixed cell wall due to that cell wall. And they are made up of the different parts.
Yes.
Now, one by one, we have to study the functions. Structure, first is the cytoplasm.
Yeah.
Function is where the chemical reactions happen. Smaller structures called the organelles can be found here. The chemical reactions occur in the cytoplasm we have studied earlier. In the keywords we have studied yesterday, that all the chemical reactions in the cells can occur in the cytoplasm.
Yes. It's
Second
like
is
where the reactions happens inside it.
cytoplasm, jelly-like
Yeah.
substance which is present in the cell.
Yes. So
Now,
it's like something like what I remember is it's like something like jelly.
yeah, jelly-like substance.
Yeah.
Now, next is cell membrane. That is the barrier around the cells and controls what goes in and out. Cell membrane is a barrier through which what will come out, what will go in, that is decided by that membrane because it's permeable or semi-permeable, how it is selectively permeable, it depends on the cell, no? Right? Nucleus, there is no written in
Right.
the function, but nucleus is the control center of the cell, and it also have the DNA, means hereditary information is there, right? And it controls all the cellular activities. All the cell
Okay.
activities
So
is controlled in by the nucleus.
okay. So like it control everything, every activity, cell
Yeah,
activity.
yeah.
Okay.
That's why it's called the control center of the cell. Understood? Now, next is mitochondria, where the respiration happens in the cell and the energy is released. Means ATP molecules are released in the mitochondria because it is the powerhouse of the cell. Mitochondria is also called the powerhouse of cell. Next is the cell wall. Are you
Is
there?
it like miss?
Next,
Is it like where it can sense?
what? Please repeat.
You said like mitochondria. Is it like where it can sense things?
Sensory things.
No, you said something like to do with sense.
I can't understand what you want to say. Mitochondria.
You just said something to do with like sense.
It's a respiration which is occur inside the cell with the help of the mitochondria. The respiration
Yeah.
process
Okay.
is done by the mitochondria in the cells, and energy is also released. So
I
that's
misunderstood,
why,
miss. Okay.
yeah. So that's why mitochondria is called the powerhouse of the cell because ATP molecules are
Oh,
produced in the mitochondria.
then. Okay.
Okay. Now next is cell wall, extra protective layer around the outside of the cell membrane, and it is made of the tough substance called the cellulose, which makes the cell firm and rigid. Understood now? firm and rigid because cellulose is
Yes,
present
miss.
there. Now next is the vacuole. That's the large bubble full of the liquid called the sap, and it keeps the cell firm and present in the middle of the plant cell, and it is filled with the liquid which is called
Yeah.
as sap. Okay, and present in the
Okay.
plant cells. Last is the chloroplast. It is also present in the leaf cells, and it is a small round green organelle, and it contains chlorophyll that helps to trap the light energy. Okay.
Okay, so it makes the light takes or like something like that.
Yeah, yeah. Now next picture, unicellular organism, which I have show you in the keywords also, amoeba, euglena, flagella. That page will come out. Okay. A unicellular organism is a living thing that is just one cell, and these organisms are very small and consist of only one cell, and they are usually found in the water. Some of these can make their own food through the photosynthesis, and
So we just
there
studied earlier miss, in the like words.
in the unicellular organisms, they are
Yes,
very
miss. You
they
told
are
me
very
about
small in the
photosynthesis.
size. They are very small in
Yeah.
size, and in the numbers it is uni means one. Unicellular means it consists of one cell only, and these unicellular organisms, they are present in the water, and these also make their own food with the photosynthesis. That is a process by which the plants can make their own food na? By the photosynthesis. It is a process. Do you know about photosynthesis?
Yes, miss. I know about photosynthesis.
Okay, okay. There are different types of the unicellular organisms, including the bacteria, protozoa, unicellular fungi, and you might be tempted to think that these organisms are very simple, but in fact, they can be very complex. They have adaptations that make them very well suited for the life in their environment. Just give me 1 minute. Yes, yes. Where we have read, yeah, you these three are the unicellular organisms like bacteria, protozoa, and the unicellular fungi. And these organisms, they are very simple because it is unicellular, na? And they also have the adaptations to suit in their environment to live. Means they are very easily accommodate with the environment, right? Next is bacteria. Bacteria are tiny. A typical bacterial cell is just a few micrometers across, and the structure of the bacterial cell is different to an animal or the plant cell. For example, they do not have a nucleus, but they may have a flagellum. This is a tail-like part of the cell that can spin, moving the cell along. Because it helps in the locomotion, flagella is for the movement of the unicellular organism, which is present in euglena, as shown in the figure. And amoeba can move with the pseudopodia with the force feed they are producing with their protruding out their body. Right? Understood? Are you there?